So you think we could start this session?
Yeah.
I guess everyone is ready. Good afternoon, everyone, and welcome to the first official masterclass of the EPIC Butterfly project. These masterclasses are part of the training program of the EPIC project that aims to strengthen the taxonomic capacity of Eastern Europe countries. This level, however, is open for everyone belonging to different countries to participate. And this is the first masterclass that will be held by Rudi Verovnik. Rudi is a professor at the University of Ljubljana in Slovenia. He's a butterfly expert with more than 30 years of experience. He's a member of the Society of Conservation and Study of Lepidoptera in Slovenia, and also runs the butterfly monitoring scheme. And Rudi has served also as a board of Butterfly Conservation Europe since 2007. He's co-author of multiple European scale studies, such as GBI, the Grassland Butterfly Indicator, Climatic Risk Atlas, Red List, etc. But also is author of many local scale studies, such as, for instance, maybe you know him because he studied the endangered Macedonian grayling, Anthocharis Sinkovsky. So I think he's the perfect person to start our series of masterclasses. And we are very grateful to have him here. This masterclass is named Eastern Continental and Balkan Butterfly Species, Anthocharis, Leptidea, Colias, and Pieridae genera. Sorry. Some technical things to add before Rudi starts is that this masterclass is going to last approximately 1 hour and a half. We will have a break in between. In between each. So we are going to have two divisions. 50% will be dividing the genera in three. We will have Pieridae, Anthocharis, and Leptidea. We will have a break, and then we will go for Pieridae genus. For you to navigate here, you can raise the hand, and then we will allow you to talk when it's doable, or you can go to the question and answers, and you can ask a question there. Every time we finish a genus, it will be time to go to the section and read the questions. Okay. If you want to ask something, we will get to you if possible, and we will allow you to talk. I think everything is set. So I'm going to allow now Rudi to go. The floor is yours, Rudi. Thank you very much, and yeah, you can start whenever you want.
Thank you, Juan. Thank you, everybody, for joining me for this first advanced course on butterfly identification. Just in case if somebody is looking at cooking masterclass, this is the wrong session. So it will be recipes only about how to determine some difficult species. The focus, as you can see, is Eastern Continental Europe and the Balkans, but some other species are also included, just to have the whole overview of the genuses. So don't be surprised to see some boreal and even West European species included. And we'll start off with something relatively simple. The Anthocharis genus, the orange tips, are a well-known group of whites, quite easy to determine, especially males, because they have, as you can see on this picture, a really large orange or reddish apical area on the forewings upper side. So it should be an easy start to go through. My presentation will be in two parts. The first part will be presentation of species, and then we'll go to identification. So let's see what I have about Anthocharis cardamines. Of course, most of you who work on butterflies know the species well, as it's widely distributed. Not really a habitat specialist, usually uses ecotones, so forest edges and different kinds of grasslands for its habitat. And it's present also on several larger Mediterranean islands as well. So it's really the most widespread of the Anthocharis species in Europe. Also not particularly choosy about its host plants, so different cruciferous, including cardamine, which is the most commonly used one, are the host plant for the species. Butterflies are usually present already in the early spring, so from March onwards, and in the higher altitudes, especially in the Alps, they can fly into June, but they develop just a single generation per year. The second species is much more localized in its distribution. It's Anthocharis damone. It's mostly distributed in the Balkan Peninsula in the southern part, but there are isolated populations also in peninsular southern part of Italy and on Sicily. In the Balkans, it has a relatively continuous distribution in Western Greece, which extends into Northern Macedonia and southern Albania. And there are some isolated locations known in Serbia. And quite recently, it was recorded also in border area of Serbia and Bosnia and Herzegovina. So it's a new country added. The habitats of the species are quite specific. They prefer more rocky areas. Depending on the region, this could be old lava flows like on Mount Etna in Sicily, or ophiolitic rocks like in parts of Serbia, or calcareous steep slopes like in most of the other parts of the range in Europe. That's also linked to the habitat of the host plant. This is Satis tinctoria, which is basically a pioneer's plant, usually growing in open areas like on screes, but also on road verges. So as a secondary habitat, road verges are also important. And adults in Europe fly from April, mid-April to end of May. As most of the locations are at lower altitudes, for example, in eastern Turkey, they can be found up to 2,500 meters, and I've seen it there in August. So altitude is an important factor in when the species appears, and it's also monoval time. Okay, next one. Anthocharis greeneri is present in Europe only in the Balkans, reaching southern Serbia and southwestern part of Bulgaria, but most of its range is in Greece. It is another habitat specialist, but this time linked to calcareous substrate, where it flies on, again, rocky slopes and screes, linked with the habitat of the host plant. So Tenemax axatile and different species of Cicimbrium all grow on open, sandy, rocky habitat. So that's basically the habitat where the species is present. It flies a little bit earlier than Anthocharis damone, with which it sometimes can be sympatric. So the first ones already appear at the end of March, and by beginning of May, they are quite rarely seen. Although in Greece, they can fly also at higher altitudes and also in May. And a Western distributed species, this is Anthocharis euphenoides. I added it also because it flies in peninsular Italy, so it's not exclusively Western. And it's the only other species of the genus present in Europe. As you see, the distribution is mostly in Iberian Peninsula and the southern France. And there are some isolated populations, or at least there were, in Western Alps, also in Italy. And in peninsular part of Italy, it's mainly linked to Apennines. The main host plants of the species is Biscutella levigata, mostly. And this is another pioneer plant which prefers open sandy habitats, rocky habitats. And this species is really typically linked to road verges where this habitat abounds, and usually you find them there, although it's in Spain it's really widespread, so it utilizes also other more natural rocky habitats and also open grasslands. The flight period is, as you can see, really long, and that's because of its range. And in southern Spain, in Andalusia, the first males can appear already in mid or end February, while in the Alps, and especially in Apennines, I've seen fresh specimens also end June, so they definitely fly also in July. But again, single generation. Okay, now let's see the differences between these species. There is no problems in determination. Of course, Anthocharis cadaminis is the most widespread and easily recognized by the fact that it's the only species in which males have white ground color on the upper side and more orange apical area. And as you can see on the underside, the other important feature, I can try to use my mouse, I hope you can see it. The green pattern is really densely structured, so it appears marmorate with green, because in other species that is less pronounced. Then all the other species, you can see, have the ground color either yellow, deep yellow, or lemon yellow. And the difference is on the underside, at least when you compare Anthocharis euphenoides and Damone. Euphenoides has the underside with quite reduced green markings, which are well defined, while Damone has a pattern which is more similar but not so dense as in Anthocharis cadaminis. And then in females, the main difference is that the tips of the forewings are orange in euphenoides, and in Damone they are grayish or blackish, quite similar to cadaminis female. And another important fact is that, as far as I know, they are not sympatric, although they fly quite close together in peninsular Italy. They don't fly in the same region and also not in the same habitat. And then Anthocharis greeneri, which is the smallest of the Anthocharis in Europe. And the ground color in males is more lemonish yellow, sometimes even less pronounced than in this case here. So it could be even paler in some specimens. And one of the characteristics I didn't write down here is that you can really see the checkered edge of the wing on the forewing in the darker part, which is less pronounced in all other species. So that's another useful characteristic you can use for determination of the species. Females are also much smaller, but they have another feature which is easy to see, and that is that the costal spot in the upper part of the cell reaches the costal edge of the wing, while in other species, as you can see, it's quite far away from its edge. So I don't think there is any difficulties here. So I will wait if there are any questions. Otherwise, we can move on to Leptidea.
Yes, I think everyone agree with you. Everything is clear. We can continue, Rudi.
Okay, thank you. Sorry. There is another thing which I have to discuss. Of course, males are easy to determine, but in females, there can be some problems. You can mix them with the following genuses, so Chloe, Zegris, and Pontia, but only if you don't look carefully, because these three genera have species with much more pointed wings, and therefore they are also much faster flyers. They usually also fly more in a direct line. And the green pattern on the underside is different, usually more defined. And of course, in males, there is no orange red spot on the forewing upper side. There is one in Zegris, but much smaller than in Anthocharis. So even in behavior, they are really different, and the wing shape should also be enough to see the difference. Okay, then to the next genus. That's Leptidea, the wood whites. Very easy group to identify to the genus level, of course, because they have really rounded wings and elongated wings, and their flight is really weak, so sort of fluttering flight. So they can be seen and determined to genus level quite easily. But when you come to species level, there are, of course, huge problems, because, as you know, in this genus, there are several cryptic species. I only present two here. There is another one, Leptidea reali, which is West European exclusively. And these species are normally not possible to identify in the field. They have to be dissected. And there are two more species present also in Eastern Europe and Southern Balkans. So we'll show how to determine those. But first, let's go through the species. Leptidea sinapis is the most widespread one. Present throughout most of Europe, including large Mediterranean islands, and it also reaches northern Scandinavia. It is, I would say, generalist, but present usually in the close proximity to woodlands or some bushy areas, and can occupy different types of grasslands, but it becomes more thermophilous towards north. So it prefers drier grasslands combination with bushes and forest towards north. The flight period is also quite long, but they have 2 or 3 generations. In the south, usually 3, in the north, just 2, which are also quite distinct. So when I will show the pictures, there is quite a big difference between spring and summer generation. Next one is Leptidea ivernica, which is, as you can see, more sparsely distributed in Europe. Interesting thing is that it's present in Ireland, but not in the UK, and vice versa, although I think Synapse was found in Ireland. It's also quite widespread in Central Europe, then isolated populations in higher altitudes or mountain areas in the Balkans, and also in southern Scandinavia and the Baltic countries have known records of the species. I think there's also a mistake on this map, because it shows that it's present also in southern France, where actually only Leptidea reali is known. So in the Western Alps and in Massif Central, I think it's just Leptidea reali. Okay, again, the species has 2 or 3 generations present from April to September. There's no real distinction in that, like, for example, in Leptidea reali, which is in most parts of its range monovoltine. And then we have Leptidea morsei, which is definitely a more East European species, which reaches its western edge in Slovenia, Croatia, and Austria, and then is present in the northern part of Pannonian Basin and Romania, and through Ukraine and southern Russia into Siberia and eastwards, because it has quite a wide range outside Europe. Of course, this map lies a little bit because the distribution is much more fragmented than shown here. And as far as I know, species is almost extinct in Austria and in Hungary, where it's, I think, only in the Aktelek cards still present. Also very rare in Slovakia and Romania. So there's basically no larger area of distribution of the species in Europe. Habitat-wise, it's more specialized than the previous 2 species, because it needs the host plants, Lathyrus niger. At least in Europe, it's linked only to this host plant. And this quite large Lathyrus grows usually at forest edges and forest clearings in deciduous woods, usually in more warmer situations, at least the parts of the range that I know, and usually in oak woods. So open oak woods are the most important habitat of morsei, at least at the western edge of its distribution. The flight period is from April to July in 2 generations. So first generation is usually from end of April to end of May, and the second one from end June to end July. Again, with pronounced differences between generations. And to finish up, Leptidea duponcelli is possibly the most easily recognizable Leptidea, at least in spring generation, with very interesting disjunct distribution. So it's present in Western Alps and southern France and in southern Balkans. So no populations known in between. And further east, of course, it's present also in Ukraine, in Crimea, and I think also in some parts of southern Russia. Again, 2 generations per year from April to August, quite distinct as well. So let's go to the identification. I have Leptidea Sinapis Uvernica as a complex here, also because in Levington's book there is no distinction between those 2 species. So I will talk about identification of these 2 species separately. I will focus now on morsei and duponcelli. Leptidea morsei is the largest of the Leptideas in Europe, quite distinctly so if you see them in nature. And it also has a little bit more prolonged, at least forewings, but also hindwings, and slightly falsate. So they have a sort of a small dent at V6, vein 6, which is better pronounced in spring generation compared to summer generation. Yeah, let me explain. So this is male spring generation, male summer generation, female spring generation, and female summer generation. So the wing shape is probably the most important characteristic, but the other one, which I find it quite useful when I'm doing fieldwork here in Slovenia, is that the hind wing underside in spring generation is more yellowish. It's not greenish like in Leptidea sinapis and Verovnika. And the central part of the wing, you usually have well-defined white spot in more grayish part of the wing. So this pattern is much more pronounced than in other Leptidea, which can fly together with it. Females are usually even larger than males, and they have more rounded wings, so this dent is not so well visible, but other characteristics are similar. The other thing which is useful is when you see the butterflies in nature, is that Leptidea morsa is the only species which can fly also in dense woodlands. So searching for host plants, the females can be seen also in deep in the woods. So that usually you don't see any other Leptidea there. And the other characteristic is that when they fly, they can also glide, which is not present in any other Leptidea in Europe. So usually that means that when they fly down, they half close the wings and then they glide down the slope. So that's something that no other Leptidea do. So it's a useful characteristics. And let's go to Leptidea duponcelli. Leptidea duponcelli is most easily recognized because the green pattern on the underside in spring generation in both sexes is really pronounced. So you have green areas and then a small white spot in the upper part of the wing, actually two such spots. So when you see that, then there is no mistake. The other characteristic is maybe not so well pronounced on these slides, is that the vein 1 on the forewing here has sort of a bump at the basal area, which is less pronounced in other species. So here you can see this is really more straight. So if you have the butterfly in the net, that's one of the characteristics you can check if you're not sure, especially in second generation, where there's no such typical green pattern. The second generation, as you can see, is more yellowish without any distinct pattern. Commonly used characteristics is also the tip of the antenna, which in duponcelli is, at least when it's fresh, it's really deep orange. So and the other part is black. While in Leptidea sinapis complex, the tip of antenna can be brownish, not really strongly orange colored, and at least the 2/3 of the antenna on the underside at least should be white. This characteristic is useful when the butterflies are fresh. When they are worn, these antennae can become uniformly brown, also not so distinctly orange in duponcelli. So only when butterflies are fresh, this characteristic should be used. Okay, let's talk about Leptidea synapis uvernica complex for a short while. If you really want to know which species you have to do the genital dissection. So that's basically mandatory, and it's much easier to do in males. So if you collect material, then it should be males, because then determination is safer and easier than in females. But there are at least Mazel, a French guy, wrote a paper about this. He showed some characteristics which might be used for determination of differentiation between males of Leptidea synapis and uvernica. Females are really variable, so it doesn't help. So the one characteristics which I show here is the shape of the dark or black area in the apex of the forewings. Inner edges of this spot are at approximately 90% angle, so the spot is really square. While in uvernica, it is more elongated, and the angle is wider than 90%. So if you see typical specimens with these characteristics, then you can say that it's possible that it's either synapis or either uvernica. Also, the underside, especially the hindwing underside, can be sometimes different in the Springer generation, and that is that in uvernica it's much more strongly suffused with gray scales, so the underlying pattern is not well visible. While in synapis, the paler parts between the veins are more pronounced, and it's more greenish than grayish on the underside. Still, I would say that to be certain, you have to collect some material and do the genital dissections to be sure what species you're dealing with at a particular location. Okay, that's it for Leptidea. If any questions, please.
All right. Excellent tips, Rudi, for this this genus that is quite frustrating sometimes when we're in the field and we cannot go with a precise identification. We have to wait for a genitalia or DNA test. We have one question from Hugo Silva, and he's asking where to find bibliography for Leptidea genitalia. I guess he means when we are doing the genitalia, where are the characters, where are the details to check and compare? So do you have any reference, either article or book for this?
Well, I don't have it out of my head, but there are several papers published on this. So if you just check the Leptidea synapis uvernica complex and genitalia, you will find the genital slides. So
okay, and do you know exactly, just maybe for everyone curiosity, where do you have to see the details to differentiate these species? What is the morphologic character that differentiate these two species in genitalia?
Yeah, well, I think there will be a special class about preparation genitalia and determination of species. So I didn't prepare that.
Oh, no. Don't worry. It's just if you know.
Yeah, I know. Yeah. It's the edagus, so the penis in Leptidea uvernica is longer, so 1.7 millimeter onwards, and the synapis is a little bit less, so 1.7 millimeters somewhere is the split between these two species. But even more useful characteristics are the shape of the valve and length of the succus, which you compare to each other, and then you get a plot where 95% of all specimens can be identified to the species level. So
excellent. Thank you very much.
It's in many papers, so it shouldn't
yes,
be easy.
We will. We have an answer from Jedrus that already pasted a link. I can look for something else also to paste in this question, but there definitely with the tips you said is enough. You just have to do the genitalia and then compare these traits. Okay, very good. I think I'm not missing any question. So.
Okay, then we go to the next
we'll
genus,
go to the
which
next
is a bit longer. The Colias genus, another really easily recognizable group of whites, which are not white usually. And there is no mistake with any other genus in Europe. But as you can see, the diversity is much bigger. You have some widespread species, some localized habitat specialist species in this group. And they have a very unpleasant characteristics. If you don't have a net, you don't see the upper side of the butterfly. So you have to catch them to see the upper side, because when they sit down, they always have the wings closed. Okay, let's see the species. This time I have split the comparisons to similar species, so it won't be identification will be also shown in between. Let's start with two species which are pale lemon yellow to almost white. One is Colias alphacariensis, which is widespread in Mediterranean part of Europe and Central Europe, but it doesn't reach Scandinavia. Also towards east, it's the range is much more fragmented. So it's known for some places in Ukraine, in the south and southern Russia, and it doesn't extend quite too far into Asia. The species is usually linked to dry calcareous grasslands because of its most important host plant, that's Hippocrepis or Hippocrepis comosa, the most commonly one used in Europe. But it can also be present in slightly different, but usually dry grasslands where Coronilla varia is present. So also places like gravels along rivers are habitat of the species because of Coronilla varia. It flies from May to October, sometimes even November, but that's just far south, in 2 or 3 generations. It's a very similar species is Colias hyale, which, as you can see, covers most of the range of Colias alphacariensis, but it doesn't extend into Iberian Peninsula, or at least there are very few records, which I'm not sure if they were ever confirmed for the higher mountains in Spain, like the Pyrenees and Cantabrian mountains. Anyway, on this map, it's that part is not shown. The species is slightly migratory, so it sometimes flies into United Kingdom, and it's also spreading north in Scandinavia. The southern part of its range is also not well defined, at least in Balkans. It's really not known how far south it goes. Some quote it also for being present in Greece. But even in Serbia, it's not present south of Danube, at least not many records are from this part. So it's basically more a Central European species, which has a range then also into Asia through Ukraine and Russia. And it has the similar flight period from May to October in 2 or 3 generations. These two species are really difficult to identify. Even if you have them in your net, the specimens, especially in females, it's almost impossible. But in males, you really have to have fresh specimens to see all the characteristics. So it's a problematic pair of species. Even DNA doesn't help that much, and there is no big difference in genitalia. So the only reliable way to distinguish these two species is the larvae. The adult larvae are really different. In Colias alphacarensis, they have black spots and a couple of orange bands, while in Hyale, the adult larva is green with just a single sideline on both sides of the body, the pale whitish line. But when you have fresh specimens, these are the characteristics that you have to look at. First one is that the outer edge of the forewing is convex, so it's protruding outwards in Colias alphacarensis, while in Colias hyale it is straight. That's one of the characteristics, and therefore also the apex of the wings is more pointed in Colias hyale and more rounded in Colias alphacarensis. The ground color is also a bit different. So in Colias hyale, not really seen on these pictures or drawings. Usually Colias hyale is a bit paler lemon yellow, while alphacarensis is sort of richer yellow color. And the other characteristic, which is also useful, but only in really fresh specimens, and of course you have to have them in your net, is the extension of the dark scaling at the base of the forewing. In Colias alphacarensis, these dark scales are usually limited to the inner edge of the forewing, so they can extend here in this direction, but they never reach the cell area. While in Colias hyale, they are more fan-like, so they are also present in the basal part of the costal cell. I usually, when I do field work and I have to determine these pieces, I check those characteristics, and if 2 out of 3 are pointing to one species, then I write down that species. Of course, there's also sort of at least in Central Europe, there's a habitat distinction. Colias hyale is usually present in more mesophilous habitats because it can utilize very different large or different fabaceae, while Colias alphacarensis is bound to more drier habitats. And Colias hyale is also present in higher altitudes. For example, in the Eastern Alps, where I know it can fly up to 2,000 meters. So usually the habitat characteristic is important to also predict what species would be flying. And then you also have a female forma helice in Colias crucea, which is somewhat similar to females of alphacarensis and hyale. But as you can see, the black outer border is much wider and longer along the edge of the hindwing. So it's really not a big problem to tell apart Colias crucea forma alba female from Colias alphacarensis and Colias hyale. Okay, Now, I don't know if we have a discussion here or we continue with other Coliases.
I think you can continue, Rudi. There is no question. I think we are good so far.
Okay, thank you. Okay, then we go to the next group of species which have a full black edge on the forewings at least. So basically, I would say Colias croquea group. And we'll start with Colias errate. Basically an Asiatic steppe species which has spread into Europe a couple of decades ago. And it has permanent colonies, especially in Pannonian Basin. It sometimes occurs also in southern Germany and southern Poland. Distribution in the southern Balkans is also a bit problematic because there are some clear misidentifications also especially from southern Greece, because you have sometimes Colias croquea which has similar coloration. But at least in Bulgaria, there are regular records in the lowlands and the foothills of the mountains. So it's present also in the Balkans. Habitat-wise, at least in the western part of its range, it is more or less bound to alfalfa fields, because these are the alfalfa plants are the most important host plants for the larvae of the species, and also it flies in steppes, which are more or less limited to Hungary and Western Romania. So the remnants of steppes are also its habitat. The flight period is from May to October. Usually each generation is stronger. So if you see the species, it's most commonly seen in the late summer or early autumn. Last year, I found it again in Slovenia after 15 years or something like that. So it obviously is still present in this region. Next species is probably the most known and most widespread of the Colias in Europe, because it's also a strong migrant. As you can see, it reaches southern Scandinavia and Baltic countries regularly. And there are also a couple of records from Iceland. So it's really a strong flyer, and it does not have permanent distribution in Central and Europe and UK, but it develops there during summer. But in the south, it can be permanently present, especially in the Mediterranean, where it also occupies most of the islands, even the smaller ones, because it's a strong flyer. Regarding the flight period, at least in my experience in southern Spain and even in Slovenia, at the coast, they fly throughout the year, so they don't have a distinct diapause, even during winter, at least if the weather is suitable. But otherwise, in Central Europe, they have 2 or 3 generations flying also in the late autumn. Colias chrysoteme is a habitat specialist distributed in Europe from East Austria, southern Slovakia, through Hungary and Romania towards east into Ukraine and southern Russia. That's a typical steppe species, linked usually to Vicia hirsuta. At least I didn't find any other reference for other species of Fabaceae as a host plant. And because it's a habitat specialist, this continuous range is, of course, not true it is really localized. I think in Austria there's just one population left, and in Hungary it's present only in the central part with these xertic steppe habitats like here near Balaton Lake. Also in Romania, as far as I know, it's just in the western part. So it is quite a localized species. It flies usually in 3 generations from May to October. And an boreal species which I added just to have the complete overview of the genus, Colias hecla. It's the only species in this part of Scandinavia. Even here towards south it is present only at higher altitudes, but of course in the most northern part of Europe it flies also down to the coast, because it prefers the low shrub tundra, also to some extent rocky parts of the tundra habitat. It's present also in the Russian part of high boreal region, reaching the polar Urals. It has only 1 generation per year, and it develops usually in mid-June, depending on weather conditions, and it can extend into August, although usually locally they fly only short period of time, and usually not in August. Okay, let's go to distinction between these species. Colias errate is identified usually by its paler coloration. So it's either lemon yellow or even greenish coloration of the upper side. And it has quite, well, I would say, medium-sized wide black border. And at least in the apical part of this black border, you can see some paler veins. But sometimes it can be also without. So if they are present just in this part of the dark border. I will not discuss females too much because they are quite variable. In Colias errate, they look to some extent similar to females of Colias hyale and Colias alphacariensis, just that this black border is usually more developed. It reaches at least the 2/3 of the outer wing on the hind wing. Okay, then you have Colias croquea, the most common species, which has a sort of orange ground color without any sheen, and the white veins in the black border are visible throughout the forewing and sometimes in the upper part of the hind wing. So using this characteristic, it's easy to distinguish. And errate, at least in Europe, it's a little bit smaller than croquea. When you see them in flight, you can see the difference. For Colias chrysoteme, there is a huge difference in size. So you can really see them when they fly, that they are something different. They have also narrower black border with even more pronounced white veins throughout both wings. And one of the characteristics which is useful also when they sit on flowers with closed wings is that the costal edge of the forewing is somewhat concave. So there is a sort of indentation in this part of the forewing, not so well visible in this drawing, maybe here a little bit better. But you can really see that when the butterflies are sitting. So it's a useful characteristic. Colias hecla is the only species up in the north, so you shouldn't have any problems in misidentification. But it's narrower. The border, the black border in the species is even narrower than in Chrysoteme, and usually full, maybe just in the upper part of the wing. The white wings are visible. Females, at least of Crocea and Derate, can be variable, so they can also be the white forms, sometimes even greenish forms. So this is quite variable. Okay, let's move on. If there's any questions, you just
Yes, Rudi, I'm
yeah.
sorry. There were two questions still
Okay.
with the two species, Iale and Alphacariensis.
Yeah. Okay.
One question that I try to answer is if there are no morphological characteristics that would help to separate the female Iale and Alphacariensis. You said no, but you can elaborate more the answer if you want.
Yeah, the females are generally problematic because they are much more variable, especially in the extent of the coloration, the black coloration, and also in the ground color. So the only thing which remains, because the also the black scaling is present in the cell in both species. So the only thing which is useful is the wing shape. The wing shape is, of course, in Hyale same as in males. So straight outer border and more pointed wings. But I prefer not to determine the females in the field.
Yes, too risky. And then you
Yeah,
mentioned
sorry. Just maybe
yes.
one remark. So if you see the female ovipositing on Hippocrepis comosa, then you know that it's Colias alphacariensis.
So
Yeah,
ecological traits more than morphological in this case.
yeah, yeah.
There is another question also related to these two species, and it's what you mentioned about the larvae. The larvae are different and could be useful to separate the species. Where to find them? They are asking, do you have any tips on finding Iale and Alphacariensis larvae? Of course, they are in different food plants, but maybe something else that could be useful for people.
Yeah. Especially for Colias africanensis, the larvae are quite easy to find on Hippocrepis comosa because it has usually quite small leaves, so you can see the feeding patterns, and then you can locate the larvae which are quite large compared to the leaves. But for Colias hyale, I have no personal experience. It's usually, well, at least in Slovenia, it's quite sparse, so finding larvae would be very difficult because they can utilize all sorts of fabaceae. Probably the easiest way to find them is to find alpha-alpha fields, because then you can find them there together with larvae of Colias errate. They also like to use this plant.
Perfect, thank you.
Okay.
That's all.
Okay, thank you. Okay, then we go to the larger Colias, which are more limited in their distribution. Colias aurorina is a predominantly Asiatic species, which flies in Europe only in southern or western Greece and in southern Albania. So a much limited range, and even within this range, it's limited to higher altitudes. So usually about 1,000 or 1,500 meters. And the habitat is quite distinct. Usually it flies on open rocky slopes where there is a lot of host plants. And these host plants are bush-like, so tiny bushes of astragalus. These astragalus were sometimes, well, in the past they were in the different genus Astracantha. So if you want to see how they look like, you can check Astracantha genus. They are quite thorny and not palatable for cows, at least. Probably goats eat them. but so they can be present also in habitats where there is some grazing going on. They fly from end of May to end of July. I would say best time is mid-June onwards to see the species in at least in the European part of its range. Colias caucasica is slightly smaller, but still quite large species compared to those I've talked about before. It has distribution which is not, or it's maybe a little bit touching the distribution of Colias aurorina, so it's present more in the northern and northeastern part of Greece, and then through East Albania, Macedonia, southern Serbia, and Bulgaria, also in Bosnia and Herzegovina, and there are at least 2 known populations also in the Croatian hinterlands of Dalmatia. So it has quite a limited distribution also outside Europe. It's only present in a few places in northeast Turkey and the Caucasus. The habitat of the species is quite variable, but in general, it's a montane species which prefers places where there's a lot of host plant. And in European part of its range, this is usually Hamecitisus hirsutus, Absinthoides also in Bulgaria. And these can grow abundantly in calcareous grasslands, but also on metamorphic rocks. So the habitat, at least appearance of the habitat, is quite different when you see these habitats on these 2 different substrates. The butterflies are on the wing from early June to end July. Again, I would say end June is the best time to search for the species. Then Colias myrmidone, which used to be one of the more widespread species in Central and Eastern Europe, but it's gone extinct in its western part, and it's now really localized in Europe. There are only a few populations left. The larger ones are in Western Romania. There are also 1 or 2 in Eastern Poland and Belarus, and then in Russia and Ukraine. I'm not sure if it's still present in Slovakia, Slovakian karst. It is definitely extinct in Hungary, Slovenia, Austria, Germany. So it's one of the species which is disappearing fast. And the reason is that they prefer open, partly overgrown grasslands and pastures with its host plant. That's usually Hamecitisus supinus, but also some other species of this genus. And these Hamecitisus usually thrive in areas which were disturbed. So in areas where they cleared the forest, or they made some roads, or not roads, but to open up the habitat, because they are also kind of pioneer plant species. And Colias myrmidone needs large areas of such suitable habitat with many localities where the host plant is growing. And I think this is the main reason why it went extinct in the western part of its range, because the habitats became too small and too isolated. This is a habitat, for example, in Romania, where the largest population is present, and doesn't look anything special. So you could say that anywhere in Central Europe they should fly if the host plant is there, but obviously they have some other requirements, possibly linked to larger areas of suitable habitat. They fly from end of May to October, usually in 3 generations, but it depends on the season. And now the characteristics. Colias aurorina is the largest of the Colias species in Europe, almost one of the largest throughout the world. And it can be easily recognized in Europe also because it has really dark orange, almost reddish ground color, with a really strongly pronounced violet sheen. So when they fly, you can really see this reflection, and you know that you're looking at Colias aurorina. When they sit down, useful characteristic is that the basal area of the forewing is not strongly orange, as in Colias caucasica, for example. And the female, regardless of the form, because you have also white form and yellow form, the underside is generally dominated by greenish scales, so it appears green on the underside. And both sexes are usually present around the host plant, so they don't fly far away from the host plants. Sometimes they do mud puddling, and then you can see them also lower down somewhere, but usually they're in the habitat. That's also true for Colias caucasica, which has the ground color more reddish orange, and it also has some shiny reflection, but not so strong as the violet one in Colias aurorina. But still, when it flies, you can see it's different from Colias croquea, which has no reflection. And compared to aurorina, it has this basal orange area well pronounced on the forewing. And Colias myrmidone, which is the smallest of the 3 species here, is the ground color is more similarly orange to Colias croquea, but again with this shiny reflection, so it can be seen in flight that it's a different species. Also, they fly a little bit faster and more straight in a straight line compared to Colias croquea. Female of Colias myrmidone is also quite similar to female of Colias croquea, but these yellow submarginal spots are complete, so they reach the inner margin of the hindwing, whereas in Colias croquea they are usually developed just 3/4 through the wing. So that's also to differentiate from Colias croquea. The underside of hindwing in Colias myrmidone is more orange, while in caucasica is yellowish. The genetic studies show that Colias caucasica and myrmidone are really young species. There's no difference in genetics between them, but they utilize different habitats, and they're not they do not co-occur. So myrmidone does not reach so far south where caucasica is present. Okay. Any questions, or we go to?
We have some questions, Rudi. Okay,
Yep.
so we have a question for Colias caucasica. Is there any new information about the distribution? In the past 10 years, the species has vanished from some localities on 2 to 3 mountains in Bulgaria, and we suppose is linked to habitat lost.
Yeah. I don't know situation in Bulgaria, but in Serbia there were several new populations discovered. So we don't really know the extent of its range, and at least in some places in southeastern Serbia, there are strong populations in habitats which are quite affected by grazing, but this grazing actually helps the host plant to grow. So the biggest problem in most of the western part of its range is overgrowing, because the host plants also disappear. But it's true, it's species which can sometimes live in really small habitat, and any change can, of course, cause the extinction of local populations. I'm not familiar with situation in Bulgaria, and specifically more in the former Yugoslav countries, and there I don't see that it's declining.
Okay. More questions now, for instance, about hybridization. Do you think Colias crocea and Myrmidone can hybridize?
There is no known hybridization.
Okay, there is no hybridization.
Because that's basically the biggest differences in this shiny reflection, which enables females to choose the right males.
Okay. And the last question before continuing is about the genetics. When you talk about genetics, are you just talking about barcoding of COI? Is that what you mean with genetics?
Well, I think they use other markers as well, so not just barcoding. But it's obviously the split between Myrmidone and Caucasica is quite recent, and this habitat segregation caused also the morphological differentiation. So although genetics can be useful, barcoding can be useful for determination of some species, it's not useful in this case.
All right. Thank you for the answers, and thank you for the questions. We can continue now with the last species of the genus.
Yeah. The last species are relatively easy to determine, so I will be really short. First is Colias ficomone, which is endemic species distributed in most of the Alps and Pyrenees, and also parts of the Asturian mountains in northwestern Spain. They are typically alpine species, preferring flower-rich grasslands interspersed with rocks. Usually they sometimes also descend lower down to feed on flower-rich meadows. So that's also sometimes you can see them also below 1,000 meters, but that's not really common. The host plants are quite different, so they utilize wide variety of fabaceae. So obviously that's not the limiting factor. They can fly from early June to August, depending on altitude. Some populations are known to occupy habitats about 2,500 meters. So, and the beginning of the flight season depends on the snow cover. And another boreal species to add to this, this is Colias tiche, which is present only in northern Scandinavia, and again, similar to Colias hecla, it flies at higher altitudes further south in Scandinavia, and also in lowlands near the coast in the far north. It flies usually in more grassy tundra and on the edges of the marshland areas. So it's less thermophilous compared to Colias hecla. And its distribution is a little bit more limited, at least in the Russian part of the range, where it only flies in small, and few populations are known only in polar Urals and in the southern part of Novaya Zemlya. While I think in Scandinavia it's even more widespread than Colias hecla. And the flight period it depends on the season, but they can start really early, already in early June, and fly into July. Usually mid-June and June is the time where you find them most commonly. And last Colias species is Colias paleno, which is a boreal-alpine species, present also in moorland and bog areas in between. The populations of those of moorland areas are endangered, but otherwise in Northern Europe, in Scandinavia, and in the Alps at higher altitudes, the species is still really widespread, and it's present almost everywhere where Vaccinium oleaginosum, the main host plant, is growing. Adults fly from mid-June to August. It depends on the habitat type. In lowland areas on bogs, they can fly already in mid-June. At higher altitudes in the Alps, still in August. Differentiation between these species, it's also really easy because they are the only greenish Colias species in Europe. Colias ficumone is the most darker green with distinct pattern of this yellowish patches, so easily recognizable in flight. Females, if you see them in flight, they can be similar to females of Colias paleno, but just in flight. When they sit down, you can see, of course, the difference. Colias tiche is the only green species of Colias in the north. It has paler gray ground coloration, or even grayish coloration of the wings, and differences also in the discoidal spot. Discoidal spot in ficumone is quite large and rounded, while in tiche it's more bar-like and less pronounced. Colias paleno, it's also relatively easy to recognize because it has the most rounded wings among all Colias species, and the dark black border is really regular without any white spots in it, and the underside is also very uniform without any marginal or submarginal spots, so easy to recognize. Okay, that's for the Colias. So any more questions?